you guys. Welcome back to studying Prescott's Microbiology. And today our topic is ribosomes, nucleoids, and plasmids. So we were talking about the inclusion bodies and the what was it again? Inclusion body and the cytoskeleton present in the cytoplasm, right? Now there are three other things that are present in the prokaryotes, specifically in the cytoplasm, and that is the ribosomes, the nucleoid, and the plasmids. Now the ribosomes and the plasmids are the essential part for the prokaryotes, for their growth, for their survival. The plasmids are not necessary for the survival, but it's kind of bonus type thing. So first we are going to study what the ribosomes are, how they are made, and what is their unit, how many parts are there, what are their functions, and how they are different from the eukaryotic ribosomes. Then we are going to talk about the nucleoid, how is it different from the nucleus, what is the nucleoid made of, is it DNA, is it RNA, and do we have a membrane around it, do we not have, can there be multiple nucleoid in a prokaryotic cell or not, and the other things, are there any exceptions to the nucleoid concept, right? Then the plasmids. Now plasmid is very interesting, and I think most people don't know what the plasmid are. Of course, those who are new to the concept of the prokaryotes or bacterial microorganisms. Now we are going to study what the plasmids are, how they are different from the chromosomes, nucleoid, like nuclear, DNA, RNA. How are they different, or are they the same things? How many types of plasmids do we have? Why do the bacteria need plasmid for? How they came into existence? So these will be the types that we will be talking about, not that much in detail, but I think a little bit enough for you to understand what the plasmids can be, what are their potentials, right? So let me start generally with telling, without reading first, to tell you what the ribosomes are. Now ribosomes is an organelle present in the cytoplasm, and I think this is the true organelle, the only true organelle present in a prokaryotic cell, right? Now ribosomes in the prokaryotes are smaller than the eukaryotic ribosomes, right? Ribosomes are made up of ribosomal RNA and proteins, and they are complex structures, right? Because even if they are one structure, they have two other structures, and we call this one structure a 50S, one part a 50S, and one part a 30S, right? This is just their structures names, and this S represents the unit they have, and that is the Svedberg's unit, right? Now before going into the structure of the ribosomes, why the ribosomes are important, what are the ribosomes? Ribosomes are the structures or the organelles present in the cell of a prokaryote or eukaryotic, of or eukaryote, that translates or translates the messenger RNA and forms the protein. Guys, protein, if you don't have protein, you will never have glucose, you will never have carbohydrates, you will never have fats, you will have nothing if you don't have protein. Everything starts from the protein, and the one that creates these proteins are ribosomes. Well, the information that for that protein comes from the DNA, but even if you have nucleus, even if you have DNA, if you don't have ribosomes, nothing will start. You need ribosomes to make proteins, and then protein will make structures. There are many structures of the protein, there are many types of protein, just as plastic can be transformed into infinity structures. So the ribosomes that makes proteins are in multiple shapes, structure, functions, and everything, right? And everyone has a different purpose. The ribosomes make that, and the protein also has different functions. Some are structural proteins, some are functional proteins, some are secretory protein, and some are just localized proteins. Now ribosomes have two types of ribosomes in the cell. One is present in the cytoplasm and one is attached to a plasma membrane, right? Plasma membrane. Now, the plasma membranic ribosomes that is attached to plasma membrane, they produce proteins that are to be secreted out of the cells, and the ribosomes that are present in the cytoplasm, they produce proteins that are needed in the cytoplasm. Now, there is another thing about the protein, that the protein needs a perfect shape for it to work, for it to be in active state. Like even if you make linear chains of the protein, of course the cores are in linear form, right? And then every 3 AGC, you can say every codon, am I going too deep? Every codon makes a different brings a different amino acid, and then when amino acids combine, it will make a chain of protein, right? Then that chain, because of their bondings and because of their polarity and non-polarity kind of stuff, they rounder themselves and put themselves into themselves and make structures. They bond themselves in a irregular. I mean, the chemistry is not irregular. I mean, they do it in a very systematic way. They make themselves in another structure because of how they are arranged in the chain. Now, how do you give a shape to a protein? Ribosomes don't usually do that. Some proteins are made directly into their final structure when they are in the process of being created, right? So they are different things. Some proteins need further modification after they are combined. Like after the amino acids combine and it forms a protein chain, it still is not in a final shape and structure. So for them to go into a perfect state, they need chaperone proteins. Wait, is it written here? So whatever, don't look at this, I don't know how to find it. So they need more proteins. They need more proteins to change them, to modify them, right? And they are known as here, they are chaperones, molecular chaperones. They bring them to that final state, right? Now, this is what we were talking about, how they make proteins, how do they structure it. I mean, this is still not the detail, but it is general. Now, how they themselves are structured. The ribosomes in size is the 70S, right? We call them 70S ribosomes. Our ribosomes are 80S. Yeah, here it is. No, I called us because we are eukaryotes, right? We have eukaryotic cells, so we are having 80S ribosomes. Then the prokaryotes have 70S ribosomes. Now, what is this S? What is the Swedberg unit? Have you heard of the centrifugation? The process where you spin a material with higher speed. This is a simpler words I can explain to you. Think of it as a dryer. You know dryer, when you put clothes and dry it. How speedily does it rotate? And because of its rotation, the water, the molecules the bonding gets weaker and the water is splashed out of the container, right? Because they have pores. But when we talk about the centrifugation, the speed is very high. 1700 rotations per minute, 2000 rotations per minute. That is the speed of a centrifuge, and even more than that, right? So the faster a particle travels within the centrifuge, the higher the Swedberg's unit is, and the faster they move, the higher their sedimentation rate will be, the higher ability they will have to sediment below, right? Sedimentation coefficient, they call that. So it's because the bigger the molecule is, the higher they will carry that force within themselves, the higher their momentum will be, the higher the gravity will be acting on them. Even though centrifuge speed doesn't allow gravity to be in motion that much, but as soon as the centrifuge stops, they are sedimented already. The centrifuge has its own gravity, you can say that. So that's why the ribosome has the 70s Svedberg's unit. When you put it in the centrifuge, this is the unit we get when we are talking about the prokaryotic ribosomes. When you talk about the eukaryotic ribosome, this is the 80s is the unit that we get. But this is not the end. We have, wait, this is mobile ringing. Yeah, stop it. So within these ribosomes, we have further two structures. They are made from two structures, and when you put them separately in a centrifuge, you get 50s for one structure and 30s for another structure. That shows that their sedimentation rate or Svedberg rate is different, that the rotation speed or the moving speed is different for both particles. This is how this looks. Now this one is 30s unit. It depends on the volume, the shape, and the density, and a lot of things. Doesn't mean if the particle is smaller, the Svedberg's unit will be smaller as well. Maybe they will have more density, but in this case, I think the density isn't that much different. So this is the 30s unit, and it is made from the ribosomal RNAs, right? Ribosomal RNA and the transfer RNA. The transfer RNA is for them to bring the amino acids. The transfer RNA brings the amino acids, and the ribosome, what do you call it? Function it, or process it to make a protein out of it, right? Now the two types of ribosomal RNA we talked about here. If you look at it here, the transfer RNA have a site where it is attached. It isn't put randomly in the ribosome. It has a site for it, right? This is, we call them as active site. It is active because it is bringing the amino acid. Newer and newer amino acids are coming because of this transfer RNA, and it is present in an active site. You're right. The site where it is present, it is active site. Now, if you look at this structure, right? This is RNA, and this one's the, what do you call this one? This one's also the ribosomal RNA, but the one is the 5S ribosomal RNA, because the structure is smaller, so it has a lower Svedberg's unit, and it is 5S. Well, if you look at the gray area, this is 23S ribosomal RNA, right? Svedberg's unit is not specific for ribosomes. It can be for any molecule, right? You put in a centrifuge. Now, if you look at the golden, it is transfer RNA, and if you look at the cyan color, whatever do you call it, like it is even more than the ribosome itself. Look at this. The protein are low in quantity, while the ribosome is not ribosome. The RNA is higher in number. And what RNA is this? This is the 16S RNA. 16S RNA. That just means that the 16S RNA is smaller in quantity than this 23S RNA. Look, the gray area is more than the cyan area. While if you look at this purple type of things or this blue type of things, these are proteins. So this is the general structure of the ribosomes, and they are saying that how did they make this structure? Ribbon diagram. They made crystals of the bacterial ribosomes, and then they exposed it to X-rays, and then they studied the diffraction patterns, and this is how the structure is made. Now the second is the nucleoid. The oid, as you can say, came from a void area, and the nucleus or nucleus material, genetic material, came from the eukaryotic side concept. Now the nucleoid will have double-stranded DNA, right? Deoxyribonucleic acid, and it can be one chromosome if they condense, right? It can be one chromosome in most of the cases, but there are cases that have more than one DNA. Now, more are a double helical structure, while some prokaryotes have it in circle shape. Some have it in a helical shape and condensed shape and make it a chromosome, while some has it in a single circular shape. The nucleoid lacks the membrane bounding, unlike the eukaryote that has a membrane delimited membrane around the genetic material and makes it a nucleus. The only difference between a nucleoid and a nucleus is that the one doesn't have a membrane, the other one has a membrane. They lack membrane because they have low area. They prefer to have the least of what the structure offers, right? Because they want to condense everything in the area. Now the nucleoid may be more than one. And this only happens when they are in cell division, like when they double their everything in a cell. So what was I talking about, man? The people here don't let me live in the PC. No, I don't know what I was talking about. Yeah, I was talking about the nucleoid. Now nucleoid we know it has DNA. Guys, remember that the bacteria don't have DNA as their genetic material. I mean, viruses have RNA as well for their genetic material, just as they have DNA, and they might have single-stranded DNA as well. But the bacteria, they lack the RNA as their genetic material. They have preferably only DNA. They have 30% RNA mixed in them, and of course 10% protein, because the nucleoid has so much DNA, and for them to condense they need something, and that the need of something, that something is that 30% RNA and 10% protein. Now if you look at this diagram, they are talking about this here. They lysed the E. coli, Escherichia coli. They lysed it, and all of its DNA nucleoid material is out. And if you look at it, it's a spring fiber shape. It is not in the chromosome shape. Look at how much it is. Tell me, how do you condense it in a bacteria that has lower what do you call this quantity? 1,004 length or shape. What do you call this? Longer? Yeah, 1,400 micrometer. And the bacteria itself is just 1 micrometer or somewhat like that. So how do you condense the 1,400 micrometer into a 1 micrometer shaped microorganism? Of course, you need to condense. And unlike eukaryotes, the proteins that we use are not histone proteins, okay? So yeah. Now, when we were talking about the membrane, right? Membrane. There are exceptions to that. And they are talking about there are two genres that we have studied till now that has membrane around it. And the phylum is the Planktomycetes, while the genre is the Prolula and the Gammeta. Obscriglobus is written here with the species, like the Gammeta is a genus while this one is a species. Now the Gammeta is surrounded by two membranes while the Prolula has single membrane, right? And the membrane has not just the nuclear material, the DNA material present in that, but also have ribosomes within that. Like, it's more like us because our nucleus has ribosomes as well. Yeah, and they are talking about the Gammeta, that we don't have enough information for that, or yeah. And even if we have that, we will be studying in the next chapter. So that's generally that the nucleoid is just a genetic material. It can be DNA, it can be in a helical structure, compact structure, in a chromosome structure, and it can be in a circular shape, right? It can be one chromosome, it can be more than one chromosome, and the condensation is done by the RNA and protein lower in number than the RNA, and both are found in the bacteria and archaea. And just that there are exceptions to that membrane property, that lack of membrane property, even the prokaryotes, right? Now the plasmids. Plasmids are the extra genetic material other than the nucleoid, other than what the nucleoid have. Plasmids can be in circular shape, can be in linear shape, right? And plasmids are not just the prokaryotic property, it can be found in the yeast and fungi that are eukaryotic cells, right? So plasmid can extend from prokaryotic level to eukaryotic level, but they are mostly found in prokaryotes and they are abundant in prokaryotes. Now, the plasmid isn't just a single entity present other than the nucleus. They can vary in number. They can be from 1 plasmid to 40 plasmid depending on the microorganism it is in and depending on the plasmid it is. If the plasmid, and some plasmid has higher copy number, some has lower copy number. By copy number I mean that they allow themselves to replicate, the ability of them allowing themselves to replicate. So the one that has 1 copy number means they only allow themselves to copy once until that copy or themselves are not degraded. So you will only find 1 plasmid at a time in that cell, or maybe 2. But if it has higher copy number, for example 40, so you will find 40 of those plasmid in that cell. No more exceeding than that 40 number, but 40 must. Now, how does the linear plasmid exist in the cytoplasm? Linear plasmid has something, properties, proteins maybe, or the coatings at the ends of the strand. Both ends of the strand has the specific DNA molecule or the proteins that allows itself to be present in the cytoplasm without being degraded, right? Usually as soon as the DNA is out and it is not modified, it is getting to be degraded. This is how it works, because why do we have a DNA that is not protected? It just means that you are degraded, right? So and plasmid are very small in shape because their genes does not code for the growth properties, growth material, and nothing else. They just have bonus properties that gives cell property to be able to survive in the environment more and more, or to function more exceedingly than the other prokaryotes. Now, are able to replicate autonomously means they replicate without needing permission from the chromosomes. They do it on their own. If they want to replicate, if they think they are needed to be replicated, they do it on their own. The nucleoid doesn't have to replicate with them, right? It is just the property of them. And now the types of the plasmids, they are episomes, just as the name suggests, epi, right? Above something or on something. So episomes are the plasmid that can be integrated into the chromosomes. Okay? And they are replicated with the chromosomes. Now, there are plasmid. When the cell division occur, how do plasmid transfer to daughter cells? The nucleoids go on and into the daughter cell equally. Not even a single gene is above or low. It is that equal distribution. But plasmid, on the other hand, don't have that mechanism. Sometimes all of the plasmid from one cell go to another and one is empty. It doesn't have plasmid, while the other hand has all of the plasmid. And sometimes some has 30 plasmid and the other one will have 10. Like the number is not, you can say, it's random how many plasmid the cell will get. It's totally random. And the ability of losing a plasmid is known as curing, right? The curing can be done spontaneously, random, or you can say randomly, or you can do it intentionally by radiations, ionizing, or mutations, or like these kind of things, right? Mutations, antibiotics, that means starvation, etc., etc. We will explain when we read it, right? Now, there are the other types, these F plasmids. F plasmid or conjugative plasmid, you call them. There are plasmids that are able to transfer themselves to other microorganisms or to other bacteria of the same strain. Remember, it's the same strain, okay? It's the same species. You don't want, they can't transfer to something that doesn't have closer, what do you call it, properties to themselves. Now, the way they transfer from their own cell to another is through a tube. They call it a pili in the scientific word, pili, right? They form a pilus and they transfer it to a cell that don't have that pilus, okay? It's because the other one lacks the plasmid that they want to help go and develop the other cell, and because of the plasmid, because plasmid has very interesting characteristics. They either provide resistance, or they either have metabolic genes that are exceeding than the growth genes that are required. They may have resistance genes to protect it from antibiotics, or etc. So it's like fitting, making themselves fittest in the environment. So the one that has the plasmid that is able to transfer to another cell, known as conjugative plasmid, it is also known as F plasmid, because the pili that they made is made from the F factor on that plasmid, so it is also known as F plasmid, and the bacteria that carries that F plasmid is known as F plus bacteria because it has it. The one that don't have that F factor carrying plasmid are known as F minus bacteria. So F plus bacteria microorganism transfer their plasmid to F minus 1, and the F minus 1 becomes F plus as well, and then further on they transfer it to other cells. This is the only type of one of the three types of the sexual reproduction in the bacteria as well. Now the other also, remember, they are also episomes. The episomes that we said that they can integrate themselves into the chromosomes of the host. So they have genes for that as well. So they are episomes as well. They are conjugative plasmid. They are F factor plasmids. They are episomes. A lot of things they have. Now we have another plasmid that is resistance plasmid, or R factor plasmid, or R plasmid in short. They have genes that are able to destroy or modify antibiotics. Okay, they have genes. They make enzymes that have the ability to destroy or modify antibiotic, and in turn saving themselves from getting destroyed. And the R plasmid have maybe a single resistant gene, or maybe as many as 8, as they have said it here. You won't believe how much annoying this resistant factor is in these days. We are eating antibiotics, we are having antibiotics, injections, syringes, everything's in our body, and we just can't get rid of these microorganisms. Why can't we get rid of these? It's because of these resistance factors. The bacteria that have this plasmid, this resistance plasmid, have ability to develop resistance against the antibiotic. That's why it is said to not eat antibiotics without your culture sensitivity test, right? Unless you are sure that this will kill the bacteria, you must not eat that antibiotic, because the only thing that will do to them is develop them, and they will develop the resistance, and then you can't do anything to get rid of them, okay? You will just go into the lab and make another antibiotic. And this is the reason, the resistance plasmid is the reason that the superbugs, they are coming into existence. Superbugs are the bacteria that have multiple antibiotic resistance. Multiple antibiotic resistance means that no antibiotic in this world is working on them. So the people that have them, they can say goodbye to their, medications, because it won't work at all. So and as soon as the bacteria know that that we have antibiotic and they have this plasmid, they will allow the R plasmid to multiply more and more and more, and they will spread this R plasmid into other bacteria as well to help them develop resistance as well. Now R plasmid themselves may have the conjugative ability or not, right? Now usually the normal way for the R plasmid to transfer themselves into another is to be a conjugative plasmid as well. They have the ability to make pillars and then transfer themselves. But if the R plasmid doesn't have the conjugative ability, it doesn't have the ability to mobilize itself, to transfer itself into another, they will simply get along with the conjugative plasmid they have and will be transferred with the conjugative plasmid to another cell, right? And in turn, even if they are not directly mobilized, they indirectly are getting mobilized because of a real mobilizable plasmid in the cell, right? And that's how they are spreading into the other species, in the other strains as well, and they develop resistance as well. You can say species as well. Now I think they transfer. The R factor is even transferred into the genres, different genres. Okay, this is no joke. Species one thing, but the genre it's getting on a higher level. Now then there are bacteria that have plasmids that kills the bacteria. Okay, their own types, they're prokaryotes, and the thing that kills the bacteria are known as bacteriocins. So these are bacteriocin producing plasmids. Now there are cool plasmids. Coal, E. coli. Something hits on your head. Coal plasmids. Coal is a shortcut for E. coli. Now these coal plasmids specifically target the E. coli and kills them. Okay, they produce cholecine. If you go into specifics of the bacteriocin, you will find a lot of different types of scenes. Scenes means killing something. So they produce cholecine, means they kill the E. coli. Now there are other as well, some other species that have bacteriocin for other species, like for example coloacin. Okay, cloacin, sorry, not coloacin. Cloacin kills the Enterobacter species. Enterobacteriaceae is a family of these Salmonella, Shigella, E. coli, Yersinia. I think these kind of all of things are coming under the Enterobacter genus, and these are their species. Now there are others as well. For example, in Pseudomonas aeruginosa, they produce pyosins, and they are located on the chromosomes. Now the plasmids necessarily are not the only thing that will have resistance genes. Our chromosomes can have antibiotic resistance genes as well, and the example is that Pseudomonas aeruginosa. Now we have other type of the plasmid. Where did it go? Yeah, here. Violence plasmid. They just bonus their own cells to be more spiky and more dangerous and more poisonous, so that it can cause more harm. Okay, violence means causing harm to something, or the factors that will have the harmful effect on the host body. So there are plasmids that have genes for increasing that violence factor, right? For example, you can say in a species of the E. coli, or you can say strain specifically, they have plasmid, this violence plasmid, and it codes for the enterotoxins. So this E. coli will be more harmful than your average E. coli, and it causes the traveler's diarrhea, right? A disease. Now metabolic plasmids. Metabolic plasmids are very nice plasmids. I'm just kidding. It's not nice. They just add more towards their daily life function. They are like a technology to us, right? A technology to us, making easier for things easy for them. So they have genes that will have enzymes that will produce enzymes that will help in a metabolic activity, or even give a cell a unique metabolic activity that the cell doesn't have before these plasmids, right? Like for example, if you take yourself a human as a bacteria, right? Human doesn't have ability to fly. Just take a plasmid that have genes for flying and put it in yourself, and then the human will be able to fly as well. Like it's adding a unique metabolic activity in that. Of course, the humans won't have that strong of a plasmid for them to fly, right? So yeah, in their case, it's not that exaggerating stuff, but for bacteria, this is exaggerating stuff. Like they have genes that carries for enzymes that can degrade aromatic compounds. It's like benzene type of, benzene is very hard to degrade, and these small microorganisms can degrade it. For example, sugar metabolysis and the rhizobium, like legume nodulation. Right, for nitrogen fixation, the bacteria that are present in the roots of the plants. Now, let me just read all of this, what I said, and then we will be done with it. Are we ready? Let's start. Wait, what do I mean by let's start? I need to bring it. Here. Ribosomes. As mentioned earlier, the cytoplasmic matrix often is packed with the ribosomes that are also maybe loosely attached to the plasma membrane. So they are saying that the ribosome have very common and very high number in the cytoplasmic matrix. That is that they mean by packed, okay? And they can be attached to the plasma membrane as well. They are not just hanging in there. Ribosomes are very complex structure made of both proteins and ribonucleic acids. They are the site of protein synthesis. Cytoplasmic ribosomes synthesize proteins destined to remain within the cells. Okay, their structure is made from the protein and RNA. Now, they are the site of protein synthesis. It's a common knowledge. Now, the cytoplasmic ribosomes synthesize proteins that are destined to remain to be within the cell, okay? While a plasma membrane ribosome make protein for the transport to the outside, secretory proteins, you can say. The newly formed polypeptide folds into the final shape. Now the protein that is being formed, polypeptide means a protein, okay? That is being formed, polypeptide, that is not still a protein, that is still not active. They go into their final shape by two things. Either as it is synthesized by the ribosome, that means either in the process of being created or shortly after the completion of protein synthesis, or just as it is being made by the ribosome, they go into a chaperones and they made it into a final state. The shape of each protein is determined by its amino acid sequence. Exactly, every protein has a different sequence, so their structure automatically changes because the amino acids, the pattern is different. Special protein called molecular chaperones are simply chaperones at the polypeptides unfolding into its proper shape. Right. They help in bonding, you can say, just as catalyze, what do you call it? Catalyst? Yeah, catalyst. No, not catalyst. Yeah, catalyst. Just as catalysts help, you can say chaperones help as well, right? Catalyst is just for different kinds of material, while the chaperone is different for proteins, specifically proteins, including a detailed treatment of ribosomes and chaperones. Yeah, we will study it in a chapter. Furthermore, yeah, because it is very tall. It is going towards the molecular biology, right? That's why it's in a different chapter. Prokaryotic ribosomes are smaller than the cytoplasmic or endoplasmic reticulum-associated ribosome of the eukaryotic cell. It's common sense prokaryotic cells are smaller. They have smaller gadgets in them. So, of course, in our case, our ribosomes are associated with the endoplasmic reticulum as well, because we need more of them. We need more. And the ribosome endoplasmic reticulum actually helps in packaging and taking them. And the endoplasmic reticulum actually has a lot of chaperones. So yeah, that's why they are attached to it directly, because they want to process it as soon as the protein, or you can say polypeptide, unfinished polypeptide gets out. Prokaryotic ribosomes are called as 70S ribosome as opposed to the 80S in the eukaryote, have dimensions about 14 to 15 nanometers by 20 nanometers. So one is the length and one is the side, okay? So they are approximately same from the both sides. A molecular weight of approximately 2.7 million. I don't know what's the unit. Do we have a unit? Molecular weight. Wait, molecular weight is in what? Do I have to search it, man? Molecular weight. Am I dumb? Do we even have molecular? Okay, Dalton. Okay. It is the relative mass of the carbon-12 atom. So it is 2.7 million Dalton. and constructor of a 50s and 30s subunit. The S in 70s and similar value stands for the Swedberg unit. This is the unit of sedimentation coefficient, a measure of sedimentation velocity in a centrifuge, right? It's a velocity at how much speed does it sediment, okay? The faster a particle travels when centrifuge, the greater its Swedberg value or sedimentation coefficient will be. The sedimentation coefficient is a function of a particle's molecular weight, volume, and shape. Now, how do you find this sedimentation of coefficient, and what does it depend on? Of course, because sedimentation coefficient is the speed, right? Is the sedimentation velocity in a centrifuge, but it depends on the weight, it depends on the volume, and it depends on the shape. Heavier and more compact particles normally have larger Swedberg's number of sedimentation or sediment faster. Exactly. The denser they are, the faster they will move. The faster they will sediment, sorry. So we have discussed the structure already, we don't have to. Now the nucleoid. Probably the most striking difference between the prokaryotes and the eukaryotes is the way in which their genetic material is packaged. It's not that striking, I mean. They just don't have a nuclear membrane. Eukaryotic cells have 2 or more chromosomes contained with a membrane delimited organelle, the nucleus. I don't say that eukaryotic cells have 2 or more. Yeah, exactly. And this is just an understatement more. We have till 43, till 74, till 80. A lot of chromosomes. In contrast, prokaryotes lack a membrane delimited nucleus. It just means membrane bounded, okay? It's not a specific word, special word. The prokaryotic chromosome is located in an irregularly shaped region called as the nucleoid. The other names are also used: nuclear body, chromatin body, nuclear region. Yeah, it doesn't matter. Usually prokaryotes contain a single circle of a double-stranded deoxyribonucleic acid DNA, but some have a linear DNA chromosome, and some, such as Vibrio cholera, Borrelia burgdorferi, the causative agent of the chlorella and cholera and Lyme disease respectively, have more than one chromosome. Usually the prokaryotes have one chromosome because that's enough for them for their growth, but yeah, stupids like that, I mean, pathogens like these, I'm just kidding. Vibrio cholera and Borrelia has more chromosomes. Both electron and light microscopic study have been important for understanding the nucleoid structure and function, especially during active cell growth and cell division. So they are saying that the nucleoid structure and function has been studied under the electron microscope and the light microscope, and specifically during cell growth and cell division. The nucleoid has fibrous appearance in the electron microscope. The fibers are definitely or probably DNA. Probably, okay? In actively growing cells, the nucleoid has projections that extends into the cytoplasmic matrix. Why is it? Because these projections contain DNA that is being actively transcribed to produce messenger RNA. Constant transcription is occurring, constant translation is occurring, and constant proteins are forming for high functions because the cell division is occurring and everything needs to be doubled. Other studies have shown that more than one nucleoid can be observed within a single cell when genetic material has been duplicated by cell division has not yet been confirmed. Yeah, this is the, what do you call it, S phase? Resting phase. By resting, means not resting. What was that phase? G phase? S phase? Oh my gosh, I completely forgot. Yeah, where everything is being metabolized and doubled. Like, look at this one nucleoid, another one, and the other one has also that kind of stuff. It is possible to isolate pure nucleoid because they are present in a single region. It's not like they are moving. Chemical analysis of purified nucleoid reveals that they are composed of about 60% DNA, 30% RNA, and 10% protein by weight. Yeah, we discussed it. In E. coli, the closed DNA circle measures approximately 1,400 micrometer, about 230 to 700 times longer than cell. Its cell is smaller. How many times? 230 to 700 times. Yet how does the bacteria DNA is present in it? because it is condensed with the help of these above. Obviously, it must be very efficiently packaged to fit within the nucleus. The DNA is looped and coiled extensively, probably with the aid of RNA and a variety of nucleoid proteins. These include condensing proteins, which are conserved in both bacteria and archaea. Conserved means that these proteins are not going anywhere, like the changes in this protein is not that much of a possibility. The more the conserved the gene is, the more the conserved protein will be. Means that code is very locked. It is resistant to changes. Unlike eukaryotes and some archaea, bacteria do not use histone proteins to package their DNA. Okay, we understand. Histone proteins are complex. They need complex machinery to be made, and of course prokaryotes don't have that machinery to make the histone protein, so that's why it is not histone. That will be the proteins that are easy to form, easy to make. There are a few exceptions to the preceding picture. What? Yeah, there is always an exception to everything. Membrane-bound DNA containing regions are present in two genera of the unusual bacterial phylum Planktomycetes. Which one? Pyralula has a single membrane that surrounds a region called the pyralosome, which contains a fibrillar nucleoid and ribosome-like particles. It has fibrillar nucleoid and ribosome-like particles. Means it isn't even ribosome looking, but they have everything in this membrane. Now the nuclear body of the gameta globus is bound by two membranes. More work will be required to determine the function of these membranes and how widespread this phenomenon is. Yeah, more study is needed. Now the plasmids. In addition to the genetic material present in the nucleoid prokaryotes, some yeast and other fungi contain extra chromosomal DNA molecules called plasmids. This is the extra DNA, RNA. Indeed, most of the bacterial and archaeal genome sequence thus far includes a plasmid, right? They are saying most of the bacteria, most of the prokaryotes have plasmids. In some cases, numerous different plasmids within a single species have been identified. So some have a lot of plasmids, some have one or two. For instance, Burgdorferi, it's not Bacillus. What was it again? No, I need to know. Yeah, Borrelia Burgdorferi carries 12 linear and 9 circular plasmids. Plasmids play more important roles in the lives of the organisms that have them. They also proved invaluable to the microbiologists and molecular geneticists in construction and transferring new genetic combination and cloning genes. It's because plasmids, you know the genetic engineering, they use the plasmids in that, you know. Chromosomes are just so unfit for the transferring of the new genetic material, so they use plasmids. And yeah, if you didn't have the genetic engineering, there are a lot of things you won't have till today. Genetic engineering, even though it's late to bloom, it's been worked so much, and I think newer and newer variety of stuff are formed. And it's all because of these plasmids. You didn't have plasmids, you would have no biogenetic engineering. Who cares? Maybe humans would have found something else, but it would have taken more time than it has taken now. Plasmids are small, double-stranded DNA molecules that exist independently of the chromosomes. Yeah, they don't need the chromosome. They are independent. Both circular and linear plasmids have been documented, but most known plasmids are circular. Yeah. Linear plasmid possesses special structure or sequences at their ends to prevent their degradation and to permit their replication. These plasmids do have a starting point where the replication starts, be it a circular or be it linear. They have sites, and the linear plasmid does have the extra material there to prevent it from being circular and the same area where the replication starts. Plasmids have relatively few genes, generally less than 30. Yeah, of course they are smaller in number. The bacteria is small as heck, and if you put plasmid larger than nucleoid, what will you have then? Their genetic information is not essential to the host, and cells that lack them usually function normally. Exactly, they are not essential for growth. However, many plasmids carry genes that confer a selective advantage to their host in certain environments. Like exceed the possible, not exceed, what do you call, they increase the survival rate of the host, but the bacteria can survive without them. It's just that having them is more advantageous for them to survive. Plasmids are able to replicate autonomously. Yeah, they don't need anyone for them to replicate. Single copy plasmids produce only one copy per host. Multi-copy plasmids may be present at a concentration of 40 or more per cells. Yeah, it depends on plasmid how much copies they want. It is in their genes. Some plasmids are able to integrate into the chromosomes after replicated with the chromosomes. Such plasmids are known as episomes. We have read this. Plasmids are inherited stably during cell division, but they are not always equally apportioned into the daughter cells and sometimes are lost. Yeah, their transferring into the daughter cell is very random. So the loss of plasmid is known as curing. It can occur spontaneously or be induced by treatment that inhibit plasmid replication but not host cell reproduction. So by the way, induced doesn't mean the artificial curing is not, it doesn't necessarily mean that humans have done it. It can be random because the bacteria is under the UV light and under the certain mutagens, right? Like for example, acridin. Thiamine starvation means it isn't getting the thiamine that it wants, and yeah, so the bacteria will use only the thiamine only for the replication of the nucleoid and will leave the plasmid because plasmid is not their first priority, right? Plasmid may be classified in terms of their mode of existence, spread, and function. Mode of existence, how do they live, right? They are circular, they are linear. Spreading means if they are movable, what do you call it? Mobilizable or not. And function, yeah, on the base of functions they have a lot of types. A brief summary of the types of the bacterial plasmid are below. Here in the table. Conjugative plasmids are of particular note because why? They have genes for the construction of hair-like structure called pili and can transfer copies of themselves to other bacteria during conjugation. Perhaps the best studied conjugative plasmid is the F factor, fertility factor of the F plasmid of E. coli. So the best studied conjugative plasmid is of the E. coli, which was the first conjugative factor to be described. That is, E. coli has been studied a lot. So you can say this plasmid is being studied as well. The F factor contains genes that direct the formation of sex pili and attach the F plus cell to an F minus cell. Okay, we gone in detail with that. Other plasmid encoded gene produce add other plasmid. Well, I think I'm skipping something. Other plasmid encoding genes products add the DNA transfer from the F plus cell to F minus cells. Okay, so F plasmid genes produce more proteins for their easiness of transferring. The F factor have several segments called the insertion sequence that are able to integrate into the host cell. So F plasmid can be integrated into the chromosome and thus the F factor is an episome. Resistance factor are another group of important plasmids. They confer antibiotic resistance on the cell that contain them. Exactly. The cells that have these resistance factors, they will have antibiotic resistance. Now, it depends on the plasmid what kind of antibiotic they are resistant to. R factors typically have genes that code for enzymes capable of destroying and modifying antibiotics. Some R plasmid have only one single resistant gene, while some has more than as 8. So, the higher the antibiotic resistant genes, the more better their resistance will be towards the antibiotic. So, the more they have, the more dangerous they get. Often the resistant genes are within the mobile genetic element called the transposons. The transposons are the part of the genetic material that can jump from this area. It can be copied. The specific area is copied solely for the purpose of being transferred into another genetic material. Usually we copy the genetic material in hope that we will transcribe it and translate it, right? To make protein, we usually decode the genetic material to make protein, but transposons can be copied for only transfer purpose, right? And thus it is possible for multiple resistant plasmid to evolve. And because of them, because they can jump here and there, so the genetic material, the genetic code they take with themselves can change the genetic code of another thing, right? Because they integrate themselves in random positions in another genetic material, so the evolution can occur. And they are usually not integrated into the host chromosome, means they are usually not episomes. R factors are of major concern to public health officials because they can spread rapidly throughout the population of the cell. This is possible for several reasons. One is that many R factors are also conjugative plasmids. That's a problem, just as I mentioned. If they are conjugative plasmid, means they can transfer themselves to other bacteria, and meaning they give antibiotic resistance to other bacteria as well. However, non-conjugative R factor can be spread to other cells if it is present in a cell that also contains a conjugative plasmid. Now, if we have a R plasmid that is not conjugative, it can be transferred to other cell if it has another conjugative plasmid. In such cell, the R factor can sometimes be transferred when the conjugative plasmid is transferred. That is, it is mobilized. Indirectly mobilization. Even more troubling is the fact that some R factors are readily transferred between species. Okay? Not strains. Between species. When the humans and other animals consume antibiotic, the growth of host bacteria with R factor is promoted. There are factors that can be transferred to more pathogenic genera such as Salmonella or Shigella, causing even greater public health problems. Salmonella and Shigella are very common bacteria. They are so common that they are everywhere, and just think of them having resistance, and think of them getting something, just in case. They are still not that much pathogenic. I mean, they do cause serious health, but think about it. If they have antibiotic resistance and somehow a virulence plasmid comes into them and make them something dangerous, even more dangerous than the coronavirus, just think about that and you will understand how the health problems are greater than you can imagine with these R plasmids. Several other important types of the plasmid have been discovered. These include bacteriocin-encoding plasmid, virulence plasmid, metabolic plasmid. Bacteriocin-encoding plasmid may give the bacteria that harbor them a competitive and advantage in the microbial world. How do they give advantage into the microbial world? Because they produce a bacteriocin, means they kill bacteria that are in their surrounding of their own types. It's because they don't have to fight for the nutrients. They kill them and takes all of the nutrients, so it's an advantage to them in the microbial world. Bacteriocins are bacterial proteins that destroy other bacteria. They usually act only against closely related strains. Because the closer related the strain is, the more competitive the strain will be. And so they don't want that, and they produce bacteriocins. And the bacteriocin, we can extract that bacteriocin and use against these bacteria in case we need it. So, this is already done. Some bacteriocins kill cells by forming... Now, how do they kill other bacteria? They form channels in the plasma membrane, thus breaching the critical selective permeability required for cell viability. They destroy their normal functioning of the plasma membrane, and everything leaks out of the cell, and outer material goes inside the cell, like no permeability at all, so the cell gets destroyed because of its not normal functioning. They also may degrade DNA or RNA or attack peptidoglycan and weaken the cell wall. Cold plasmid contain genes for the synthesis of bacteriocin known as colicin, which are directed against the E. coli. As the name suggests, the colicin against E. coli, and sin means killing. Other plasmids carry genes for the bacteriocin against other species. For example, chloracin kill Enterobacter species. Some cold plasmids are conjugative and carrier resistance gene. We already said that R plasmid can be conjugative. So coal is not that much special. It is a R plasmid. It should be noted that not all bacteriocin genes are on plasmid. Oh, sorry, it's not an R gene. I was getting confused. So they are saying that the bacteriocin genes are not on plasmid. The resistant genes are not necessarily on plasmid, and the bacteriocin genes are not necessarily on plasmid, but they are more commonly found on plasmid. It can be on chromosomes. For example, bacteriocin genes of the Pseudomonas aeruginosa, which code for proteins called pyrosin, are located on the chromosomes. Bacteriocins produced by the normal flora of humans and other animals also are components of our defense against invading pathogens. We also have bacteriocins that kills bacteria because we don't want extra bacteria in our body that we don't need. Violence plasmid encode factors that make their host more pathogenic. For example, enterotoxigenic strain of E. coli cause traveler's diarrhea because they contain a plasmid that codes for enterotoxin. Metabolic plasmid carry genes for enzymes that degrade substances such as aromatic compound toluene. Metabolic plasmid just have enzymes for further functioning. So they have enzymes, they have genes that degrades what? Aromatic compounds, for example, toluene. Pesticides means the pesticide won't work on them again. Sugars, they degrade lactose. Metabolic plasmids even carry the genes required for some strains of rhizobium to induce the legume nodulation and carry out nitrogen fixation. So the nitrogen fixation is not only on chromosome but can also be on the plasmid. Now looking at here are given just example that if you talk about a general classification of the plasmid, what types are present in that, what are the examples, you can say, what are their size, what are their copy numbers, like how many can they be found in a cell, and what are their hosts, and what are the phenotypic features, what phenotypic features they exhibit. So in fertility factor they have given there is not type of fertility factor, it just it is transferred. Its size is 95 to 100. It's quite big for a plasmid, not that much big, average. Copy number 1, 2, 3. Host E. coli, Salmonella, Citrobacter. So E. coli do have, by the way, not all of the E. coli have F plasmid, okay? So if they have F plasmid, means conjugation will occur and obviously sex pairs will be formed. In R plasmid we have RP4, R1, R6, R100, PSH6, PSJ23A, a lot of that. I don't know if I have to go into detail. Well, just for fun purposes, we should go see it. RP4, for example, Pseudomonas and many other gram-negative. R1 is gram-negative. R6, E. coli, Proteus mirabilis. What is this phenotypic characteristics? This is definitely R plasmid, and so this R plasmid